A lot for the introduction and for the invitation to present here. So Juri and I are part of a team of people who are starting this Understudied Proteins initiative. And so over the next half hour or so, we'll aim to outline what we think the problem is and how it could be addressed, and also how others could get involved, because we are still very much in the beginning of this project. Of course, there is a clear overlap with the Illuminating the Druggable Genome program, but I think there are also differences. For example, as you will see, we're not focusing on druggable genes specifically, but rather aiming at a broad molecular characterization of the proteome. And also, most people of our team are, at least currently, come from a proteomics background, and that, of course, defines our technological approach for now. So let me just get started with a joke, an old joke, actually. So this is about a drunk guy who is looking for his wallet under a streetlight, and a policeman comes along and starts to help him search, but after a while they still haven't found it, and the policeman asks, "Are you sure this is where you lost your wallet?" And the drunk man says, "No, actually I lost it in the park, but this is where the light is." And so it's an old joke because in many different shapes and forms it has appeared over centuries in different cultures, and it refers to a very common type of observational bias, which, as you know, also affects scientific research. Essentially, it boils down to the problem that the best place to look for something isn't always the best place to find it. And in biology specifically, this streetlight effect manifests itself among others, in the existence and in the persistence of understudied proteins. And this is a phenomenon that was shown by multiple studies. Some of the key studies are shown down here. Essentially, the big problem is that there is a massive bias in the functional characterization of the human proteome. So some proteins, like p53, are extremely well analyzed. So there is on average 2 publications every single day involving p53. But at the same time, there is thousands of human proteins or genes on which there are no publications yet, or barely any publications yet. More systematically put, at the moment, around 95% of all publications in the life sciences, they focus on a small subset of only 5,000 human proteins, so out of at least 20,000 human proteins that we know of at the moment. So clearly that's a massive bias. And it was originally believed that the sequencing of the human genome would help to mitigate this bias. But in 2011, so 10 years after the genome was published, 75% of publications still focused on genes that had been known before the genome was sequenced. So people haven't really switched to newly discovered genes in their investigations. And clearly this bias is present in humans, but it's worse in model organisms such as yeast and Drosophila, and it's even much worse in non-model organisms, and those are often the ones that matter most in a clinical sense, such as pathogenic bacteria and fungi. So one could say that perhaps this is this bias simply reflects the fact that understudied proteins are those that don't really matter, or that are less important. But we know now from genome scale, sort of more unbiased experiments, we know that this is simply not the case. So just to give you a few examples to illustrate that point. In this publication, Wang et al. They found that out of all the genes that were essential for the growth of a human cell line, 18% they classified as uncharacterized. And more striking in this study from Craig Wenter's group, where they published the synthetic minimal organism, so a bacterium that had the genome engineered to function with a minimal set of genes possible. They said that almost a third of those genes turn out to have known molecular function. And more recently, we saw the same thing or similar thing with the COVID literature. For example, out of many human host genes that were identified in GWAS studies, more than 50% were not pursued further in targeted studies. So so just propagating this sort of bias. Of course, there are also undecided proteins are also not or are also important clinically. For example, in rare diseases, we know many genes involved, proteins involved in rare diseases are poorly understood, but rare diseases aren't rare collectively speaking. Equally, in more common diseases such as cancer and neurodevelopmental disorders, we know now that they're often driven by rare genetic variants which are less well studied. And of course, as you know better than me, the Illuminating the Druggable Genome program has reported that only 5 to 10% of the genes thought to be druggable are actually, or the proteins thought to be druggable are actually being targeted by FDA-approved drugs, at least in 2018. And finally, all of this is presumably just the tip of an iceberg, because many human proteins are still being discovered at the moment. So this paper down here, for example, reported just half a year ago, 7,000 new human genes. So that was a consortium of that involved Ensemble and UniProt. And the reason these type of genes were undiscovered before is simply because they were too small. So they're largely small open reading frames which encode microproteins. So what could be the reasons for why proteins are or remain understudied? Again, there is literature about that. And so we know there's at least two broad categories of explanation, to broad classes. One has to do with practical considerations and one has to do with the research culture. Practical considerations involve, for example, the abundance or the solubility of a protein. Of course, if a protein is more abundant, it's easier to detect. If it's more soluble, it's easier to manipulate biochemically. And also one of the most striking features of in terms of explaining how well characterized a protein is its size. So this figure is from a paper that Juri and I published a few years ago, where we just grouped human proteins, by their UniProt annotation score, which reflects how much information is available in UniProt about these proteins. 5 means these are very well characterized proteins. 1 means they're very poorly characterized. And if you look at the proteins that are below 15 kilodaltons, so quite small proteins, they make up a very small percentage of the best characterized proteins, but they make up almost 40% of the least well characterized proteins. And this doesn't even take into account the existence of the new microproteins that are being discovered now. Another interesting aspect of this is that as a way to standardize research conditions, we tend to study cell lines and yeast, for example, in the same type of media. And the proteins that are not expressed in those media or in those conditions, or that are not functional for some reason in those conditions, they tend to fall under the radar. A very nice illustration of this is the yeast knockout collection, where we know that in a standard growth medium, about 20% of the deletions give a lethal phenotype. However, if you expand the condition range and you test a wide range of growth conditions, it turns out that 97% of all yeast genes actually do have some growth phenotype. Similarly, of course, the amount of knowledge that is already available about the gene, such as if there's a phenotype or if you know about protein-protein interactions, will have an impact on how easily these genes are being studiable and how much they're actually being studied. And the availability of tools such as antibodies and plasmids is also known to play a role in this. The second part of the second type of consideration is a little bit more abstract, and it has to do with the research culture. So genes and fields that are more popular in the sense that more people are working on them, they tend to generate papers with more citations, which tends to generate more funding and increase academic success. So that's one well-known fact. What is less well studied is that often, at least funding agencies seem to traditionally appreciate more hypothesis-driven research rather than question-driven research. And it's very difficult to formulate a research hypothesis on a protein if you have absolutely no information to begin with about this protein. So that could also contribute to genes remaining understudied. And so all of this is basically summarized in this simple cartoon here. Basically, if you have tools and information, data, publications about the protein, this can help you to generate more experiments and discover the function and its potential role in a disease more easily. However, if you have a protein where there's very little or almost nothing known and you have no tools, then it's really difficult to get off the ground and to start actually approach these proteins in a scientific study. And this is where we think the Understudied Proteins Initiative could come in. Very broadly speaking, by somehow providing a basic molecular characterization, such as protein-protein interactions, for as many human proteins as possible, and thereby reducing this obstacle, this entry point of proteins into scientific research. And so with that, I will hand over to Juri, who will tell you more about the initiative itself.
Indeed. Good. So we thought about how could we possibly best and in a scientific manner tackle this challenge, and came back with that each of the people we spoke to, including of course ourselves, had very clear ideas and expectations what should be taken as like a solution to this problem. However, we also realized that, well, we sample a very small group of people and we hold our own biases. So we thought, why don't we step back a little bit from the problem and try to define it first a little bit better and get also an idea about the problem space, and then develop subsequently solutions collectively. And in order to do this first part, we established a survey that we posted, and you have here the link to the survey. And by all means, please take a moment and scan it with your mobile phone. Maybe you can, while the talk is boring, fill in the survey. It's actually very short and very quick. Or you do it afterwards, maybe when you're waiting on the bus, or have a minute to spare. And what is it that we want to answer through this survey? So let me tell you. First is we want to ask, what is the minimal information and tools for protein to be tractable? Georg was already presenting some ideas about information and tools that seem to be important from the publications of other people, and it does sound sensible to us as well. But can we underpin this in a kind of like people's scientist way? The second question is how many proteins are apparently untractable? Depending who you ask, different numbers come back because different measures are taken. And we thought that for the purpose of this initiative, we should develop our own, which of course links to the first and kind of like gives a feedback loop. Because if we look back from the answers of the people on what information they require, then at the same time that should should hold true when we look at the proteins that are labeled as intractable. And finally, where do people, biologists actually look for the functional annotation of proteins? Because whatever we find out new, we probably want to put them at those places so that it's easily picked up. Just to explain you what does the survey actually do, it asks you to annotate the annotation level of a protein. So you're not supposed to annotate the protein, you're just supposing you get the protein, you have a look at it, and you use whatever resource you normally would do in order to state whether this protein, this particular protein, is really well studied and annotated, or whether it's so-so, or whether, whoa, no idea what it does. And by doing this many times, and many people doing this repetitively, so that we have multiple votings for every protein, so we can get out the bias of individual people as well. We hope to create a human-curated set of current intractable proteins. And from there, we can then back calculate what is the information that these proteins lack that other proteins hold. And we can, over time, also monitor the progress of how maybe different measures that we or others may undertake change this problem setting. So the survey is online. By all means, please go there. Keep in mind that we don't seek the solution right away. We're kind of like working on first defining the problem as well as possible in order to have the basis to then start to come together and discuss what the solution could be. And we have a couple of people already involved. All of these have to do with proteomics. That's our bias. But I mean, it's we have defined kind of like the scope of our initiative in a white paper. This survey is ongoing, and then we want to have a workshop somewhere later this year or maybe early next year, which is meant to be an open workshop. And not everybody on Earth will be interested in joining, but whoever is interested in joining, we want to talk together with and work out together a possible way forward here. So we note the stage one idea, the survey, which is online. Now we're looking at all the human proteins. We have 20,000 of those, ignoring all the newly appearing ones for now, which, I mean, as they are just being discovered, can probably safely be put in the understudied category straight away, but they will also not be very informative because, I mean, really not much is known about them, except that they probably exist. And by help of you guys and hopefully many other scientists, the 20,000 human proteins will be sorted into like well annotated, reasonably well annotated, and poorly annotated. And based on this, we can then hopefully also work out the machine learning sorting algorithm that scores these proteins automatically in future, so that we won't have to redo this exercise every year in order to see how the annotation level changes over time. And possibly we could even go back in time and take snapshots of annotation from past years and have a look at what in the past actually raised the annotation levels substantially. So we could possibly, in a data exploration way, find out what are, in fact, valuable approaches to address it. My hope is, of course, it's my personal favorite tool, whatever it is, but the data will tell us. That's the idea. Good. So once we have the outcome, then the idea is to have this workshop, and there's at least 6 different areas that we have identified that need to be discussed. They cover data generation, of course. Hopefully, based on the data from the survey, we get a better idea which tools seem to really help. But I mean, this all needs to be discussed. How the results are to be disseminated. Again, when people in the survey state shortly where have they actually looked for this information. Then we get back the information, where should we place novel information. We will have to discuss what models to address and what conditions, how data integration may work, how we assess the progress of our initiative, which may link intricately to this machine learning based score, so that we can track annotation levels of large numbers of proteins. And no, of course, not least of all, have some sort of quality control surveillance, so that we know that whatever information we're adding is actually high quality or useful information, and not adding to the general noise to be found in the internet. There is, of course, many initiatives, including your own. We think that we are different, as Georg already said at the onset. However, there is an interest overlap, and that's why we also seek alignment and hope that we can gain and mutually benefit from our initiatives. Good. So home office, so then the kids come in. Our technological first step is protein-based, because it is proteins that we are interested in and their function. So we have at the starting point where we still are right now, a heavy bias towards proteomics technologies. But we also think that because proteins are at the center of the interest, that's a reasonable starting point. And also proteomics has come quite a long way recently in getting actually quite a diverse aspect of functional information about proteins. Localization, interaction, even functional aspects can be screened in large numbers. And of course, we cannot work up one protein at a time. So this cannot be our objective. It has to be large-scale data. And the endpoint will not be fully and finally annotated proteins. No, it's about the minimal annotation level, such that molecular biologists or cell biologists feel inclined to pick up a protein that they may find out in a different way in a screen, that they find this actually a tractable protein, and not dismiss it saying like, wow, nothing is known about this protein, there's no tools, it's suicidal for me to now start working on this protein. It's a way too high risk. And maybe at this point, I hand over back to Georg, and he can explain you about one of the proteomics tools, and then I explain you about another one. And they happen to be, of course, our personal favorites. But just to give you an idea of what is the type of annotation data that proteomics can generate.
Thanks. Yeah. So one tool that we're trying to use in my lab is protein covariation analysis. And so this is actually from a paper that we made back when I was a postdoc in Juri's group. So we published this database called Proteome HD, which was a catalog of protein abundance changes in response to various biological perturbations. And these perturbations included anything from drug treatments, differentiation, looking at different cell types, comparing different cell types even. And it was made at that time exclusively by reprocessing proteomics data that were already in the public domain. Although nowadays there are high throughput proteomics platforms where you can actually generate now quite large proteomics data sets experimentally from scratch. In any case, no matter how you produce the data, the basic principles of how you go from the protein expression to function prediction is always the same. And you see that in this figure here, that's 15 different perturbations in Proteome HD and 2 types of proteins. So those that belong to either oxidative phosphorylation or ribosomal RNA processing. And I think you can appreciate that these 2 functionally different groups of proteins respond to these perturbations in a quite different but distinct or defined way. And so the idea is very simple. If you can capture and compare these kind of patterns, you can make predictions about which proteins have similar functions. And next, you can look at understudied proteins and try and associate them in a Gilbert association approach with better studied or well-studied proteins. And you could look at these patterns using correlation analysis, but you could also, or what we have done is use unsupervised machine learning. And on the next slide, you see we basically produced out of this database a covariation map of the human proteome. So this is, it could have been a network, but in this case, a network looks like a hairball because you have so many different connections. So we rather display it using t-SNE, which is an algorithm, a dimensionality reduction algorithm. And in this case, the proximity between these proteins in the map basically reflects how similar they responded to the perturbations. And even though this was, we're just looking at protein covariation and not physical interaction, the map essentially, as you can see on the next slide, the map reflects the physical organization of the cell. So you have nucleolar proteins grouped together and ribosomal proteins and mitochondrial proteins grouped together and so on and so forth. In this case, for validation, we didn't pick a completely understudied protein, but a protein that had actually been fairly well studied, called PEX11 beta, except that PEX11 was thought to be, or is known to be a peroxisomal protein, but in the covariation analysis, it looked like it was strongly covarying with various proteins of the mitochondrial, basically of the inner mitochondrial membrane respiratory chain proteins. And so we teamed up with an expert in peroxisome biology, Michael Schrader from the University of Exeter in the UK. And when he overexpressed PEX11 in human cells, so here peroxisomes are shown in green, you can see that it doesn't localize directly to mitochondria, but it drives the formation of protrusions from these peroxisomes that contact mitochondria. And so even though these, it's not a physical interaction, it appears that the PEX11 does link peroxisomes to mitochondria. And we don't know exactly what the purpose of this is, biologically speaking, but obviously mitochondria produce oxygen radicals, and peroxisomes have many antioxidant molecules. So it seems likely that whenever you increase respiration and when you upregulate mitochondria, it makes sense to perhaps increase the delivery of antioxidant enzymes in biomechanism involving PEX11. And so, yeah, so that was just a brief introduction to what we think we can do with sort of protein covariation analysis. And then I think Juri will show you another approach.
Indeed, another technology, again, protein-based, but this time using chemical reagents in order to link proximal proteins together or internally, by the help of chemical agents called crosslinkers, that allows us then to read out in a proteomics experiment these linkage points that bring the two or that are spelling out in the native fold and even possibly in the cell, because the crossing can also be done in intact cells. Which proteins were near to each other and in which orientation even, and with help of AlphaFold, possibly this could even go into the direction of getting in a high throughput way protein interactions at pseudo-atomic resolution models, of high quality pseudo-atomic resolution models. And why that is very useful, I want to show you in an example where we in fact added these crosslinkers to whole cells, bacterial cells, in this case Bacillus subtilis, and found quite a surprising number of proteins with currently unknown function in the very small network that we generated. So this is not just a few hundred protein interactions that we detected here in this way, and nobody knows how many protein interactions exist in a bacterial cell. So also we don't know, so we don't know what is actually the target that one should reach. But there's very probably quite a lot more than what we detected here. So we just detect the tip of the iceberg, and there's a very heavy bias here towards very abundant proteins. And you should, of course, think that that plays also a role in what we know already about the interactions of proteins in these cells. And that's why I was at least very surprised when I saw that there is really, even among this tip of the iceberg proteins that we see here in interactions, quite a number of proteins with uncharacterized function. And the reason for that, so we believe at least, is that we cross-linked the cells. So we didn't investigate protein interactions after lysing cells, but before lysing the cells, because we fixed them, and this fixation we read out. So after they are cross-linked, you can do whatever. In fact, we digest the proteins into peptides, we make the peptides fly in the gas phase, and we smash them to bits and pieces in the mass spectrometer to convert them into digital signals. So this is, it doesn't need to be native any longer. The moment we conserve the information is the critical moment, and then we only read out, and we read out in a totally destructive manner. Of course, good. So we looked in closer detail here in our study at one interaction, where an currently uncharacterized protein was found to cross-link with two members of the pyruvate dehydrogenase, which is like one of the best studies enzyme complexes that we know of. And it's kind of like surprising that, well, in this study we find an uncharacterized protein interacting with that. And in fact, it didn't when we lysed the cells just straight away without cross-linking first. And this currently, or this previously uncharacterized protein, eluded in size exclusion chromatography, where proteins that interact should elute together, and left is larger complexes, right is here smaller, or even free protein. And this protein like eluted very heavily in the free protein fraction and not with the other subunits of the pyruvate dehydrogenase, or PDH. Whereas when we cross-linked in cells and redid the exact same experiment, lysing cells and running the protein complexes over size exclusion chromatography, then you see that it very nicely co-migrates. So through cross-linking in cells, we capture interactions that are lost upon lysis of the cells. Now, when we when we added these proteins into AlphaFold, and I don't know how aware you are, this is one way of looking at the quality of an AlphaFold model. This is giving you the expected error in the positioning of the amino acids in the resulting interaction model, in this case of PDHA and this uncharacterized protein. Then there is a very nice model, so low error. No, that's good. High error is obviously poor model quality in that area. High model quality in one protein, high model quality in the other, and a high model quality for the interaction of the two. So AlphaFold tells us it can make a really nice model of these two. However, when you look at the other protein that we saw cross-linked here with YneR, then the model quality is very poor for the complex. Now that we know based on the cross-links that the two are in fact in a complex, we can add this to this model and recalculate, and in fact then it all looks perfect, and that's something that one could do exhaustively by AlphaFold. You could just do all kinds of random pairs. However, it would be tremendously resource taking and possibly taking quite longer than you can spare to do that. So based on the cross-link information, we can use this information to target the bonding process. We have one other thing is in that we know exactly like not only which proteins, but we know which residues are near to each other. And that helped us in this case, even with model selection, because it wasn't the top ranked model that was the one that fit to the experimental data. It was the 7th ranked model. So their model selection played a role, or cross-link helped in model selection. And the model was a straight shortcut to the biological function, because this protein sits right on top of the binding pocket of pyruvate. And here you can see this protein. In fact, we renamed it here already to PDHI, to inhibitor of PDH, because when this slides in here, there is no access of pyruvate to the binding pocket any longer. And we did make pointments where we mutated this residue, and those completely cannot bind. And in fact, they don't abolish the pyruvate reactivity or PDH reactivity in a growth assay. Here you have the growth assay grows over time when we feed the cells pyruvate. When you overexpress YneR, then they grow very poorly. Whereas with deletion, and in fact also with the mutants, sorry, I don't have them in here, but it looks exactly the same. Also with the mutant being overexpressed, the growth is fine. And this is not a general growth phenotype. If you do feed them glucose, then it doesn't matter if you overexpress YneR or not. So really from not knowing what this protein does to having a very clear idea. Of course, there's loads of aspects that we don't know now about this protein. How is it regulated? Why is it there? How is it useful to the cells? We don't know, but that is then what we would say is for future targeted molecular biology studies to be found out. All right. With this, I want to remind you of the presence of the survey. Please go there. It's for each protein that it takes something like a minute or two, and you're welcome to do as many as your waiting for the bus takes you. And we are also welcoming questions at this point, of course.